DNA, deoxyribonucleic acid, which is genetic material. DNA is a molecule that is present in all living cells and that contains the information that determines the traits that a living thing inherits and needs to live. It is made up of subunits called nucleotides. Adenine, A-D-E-N-I-N-E. Thymine, T-H-Y-M-I-N-E. Guanine, G-U-A-N-I-N-E. And cytosine, C-Y-T-O-S-I-N-E. 1950s biochemist Erwin Chargaff came up with the number of adenine always equals the number of thymine. The number of guanine always equals the number of cytosine. This is Chargaff's rule. Rosalind Franklin, X-ray diffraction on DNA. Based on her results, DNA has a spiral shape. Watson and Crick used Franklin's info and Chargaff's rule and concluded DNA has to be a spiral ladder or a double helix. A chromatin is material that in cells that contains DNA and carries genetic information. A gene is a string or sequence of nucleotide bases that codes for a specific trait or instruction. A genotype is a specific genetic makeup. A phenotype is a physical characteristic. A chromosome is a structure of protein and DNA condensed when the cell is ready to divide. Every cell has 46 chromosomes or 23 pairs. RNA is ribonucleic acid, a molecule that is present in all living cells and that plays a critical role in protein reproduction. It is made in the nucleus and is a copy of DNA. It is different from DNA because it has one more oxygen, it is single-stranded, and it has 4 nucleotide bases. But the difference between the nucleotide bases is instead of having thymine, it has uracil. U-R-A-C-I-L. To make RNA from DNA, you have to have rRNA, which is ribosome RNA, mRNA, messenger RNA, tRNA, which is transfer RNA. Step 1. DNA gets unzipped. Step 3. mRNA carries the message out of the nucleus into the cytoplasm. Step 4. mRNA attaches to ribosome. Ribosome moves along the RNA and reads each 3-letter code.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules. It's similar to copying information on a computer. Hundreds of megabytes of data can be backed up in minutes by burning a copy onto a CD. The copying process is accurate, typically less than 1 mistake in a billion. The DNA copying process in our bodies is even more accurate, 1,000 times better than most computers. Remarkable biochemical systems have evolved to double-check the new DNA strand and fix any mistakes.
The cell cycle is the life cycle of a cell that includes cell growth and cell division. The first step is interphase, the phase in which the cell grows into its mature size and makes a copy of its DNA and prepares to divide into 2 cells. Step 2 is mitosis, a stage in which one copy of DNA is distributed to each of 2 daughter cells. Mitosis has 4 steps. The The first is prophase. In prophase, the chromatin in the nucleus condenses to form chromosomes. The centriole forms spindle fibers, and the nuclear membrane breaks down. Step 2 is metaphase. The chromosomes line up across the center of the cell. Spindle fibers attach to the centromere in the chromosomes. Step 3 is anaphase. Chromatids split. Chromatids travel to opposite ends of the cell, and the cells stretch out as the opposite ends pull apart. The fourth step is telophase. The chromosomes begin to lose their roll-like appearance, and the nuclear membrane begins to form. The third step in the cell cycle is cytokinesis. The cell membrane begins to pinch off. Two daughter cells are formed, and each has identical DNA, half of the organelles and half of the cytoplasm. The two daughter cells then begin to start interphase again, and the whole process repeats itself.